Hello, everyone. In this project, I'll be analyzing a prostate cancer DNA microarray dataset, and the objective is basically to classify patients as cancer or non-cancer based on gene expression measurements. To begin with, the dataset contains about 79 observation and 500 genes expression variable, along with a binary response variable, 1 or 0. We can see that this is a very high dimensional data since the predictor variable are much larger than the sample size. Then, as we can see from this visualization, the two class labels are fairly balanced. Next, I apply the Kruskal-Wallis test to each of the genes individually, and basically Kruskal-Wallis test measures how strongly a single predictor is associated with the response. And looking at the test statistics, you can observe how the range is, and we can conclude that for the top 9 powerful predictors to have this kind of test statistics, that means majority of the other features would have little or weak association, meaning that most of the variables are not informative at all for classification. Now, looking at the box plot also, we can observe the same relationship we saw in the Kruskal-Wallis test statistics. For the strongest genes, as we can see, the median level for class 1 is larger than that of class 0, which indicates very strong discriminative power. Also, we have the 9 weakest associated variables, and we can clearly see that the median level for class 1 and class 0 are very low. Now, moving on, I examined the correlation between all the predictors. Looking at these plots involving all the predictors variables, it practically makes no sense because it's unreadable because of the dimensionality of the data. And therefore, I considered smaller subset to see the correlation among the genes, which we have considered earlier, the 9 strongest, the 9 weakest, and just the others. Also, now looking at the again decomposition of the correlation matrix, and we observe that the ratio between the largest and the smallest eigenvalue is very, very large. You can see times 10 raised to the power 15, which means severe ill conditioning. It simply means that there is a lot of redundancy in the data. Now, after that, I trained the 6 learning machines, the 1NN, 7NN, 9NN, the 3 CPU of 0, 3 CPU of 0.05, 3 CPU of 0.1, and I compare their predictive performance using this ROC curve. And according to this curve, we can see that the 1NN classifier produced the outmost ROC curve, as you can see, green, indicating it's the one with the strongest discriminative power. And then we have the tree of CP equals to 0, then other trees follow. Then we can also observe that the 7NN and the 9NN classifier were the ones that apparently did not perform so well, they performed so badly. Next, I compare the tree by varying their complexity from 0 to 0.05 and then to 0.1. Now, changing the tree CP from 0 to 0.05 made one of the branches to be shared. In machine learning, it refers to as pruning. Then after I changed the CP value to 0.1, but unfortunately there was no change at all, meaning that no pruning occurred. And then we can interpret that beyond that CP value, no further pruning occurred because only limited number of splits provided sufficient improvements to justify, the variables inclusion. Now, the next thing was that I considered the ANOVA, the analysis of variance. And then basically ANOVA compares the mean test error of the 6 learning machines. And the statistics is 6.739. The p-value is 4.02 times 10 raised to the power 6, which is extremely small. That means that the observed difference in average test error across the learning machines are very unlikely to be due to random variations alone. And summarily, the main lesson from this analysis is that when the number of predictors greatly exceeds the sample size, performance depends less on using all available variables and depend more on identifying relative structure. Thank you.